Everyone, welcome back to my YouTube channel, and today we are going to talk about identification of bacteria. So let's start topic with introduction. Identification of bacteria is a fundamental aspect of microbiology that involves determining the genus and species of bacterial isolate. Accurate bacterial identification is essential for diagnosis of infectious diseases, selection of appropriate antimicrobial therapy, epidemiological studies, and quality control in food and pharmaceutical industries. Bacterial identification is based on combination of morphological, cultural, biochemical, antigenic, and molecular characteristics. Importance of bacterial identification. Bacterial identification helps in understanding the cause of infection and guiding effective treatment. It assists clinicians in choosing suitable antibiotics and preventing the spread of infectious disease. In research and industry, identification is crucial for studying microbial behavior, ensuring product safety, and maintaining quality standards. Morphological methods of identification. Morphological characteristics are the earliest and simple criteria used for bacterial identification. Microscopic appearance. Bacteria are examined under a microscope to observe their size, shape, and arrangement. Common bacterial shapes include cocci, bacilli, and spiral forms. Arrangement patterns such as chains, clusters, spheres, or palisades provide useful diagnostic clues. Sustaining reactions. Staining technique help differentiate bacteria and highlight structural features. Gram staining is the most important differential staining method and classify bacteria as gram positive or gram negative based on cell wall structures. Other stains such as acid fast stain, spore stain, and capsule stain aid in identifying specific groups of bacteria. Cultural characteristics. Cultural characteristics refer to the growth pattern of bacteria on artificial cultural media. They include colony morphology and growth requirements. When growth on solid media, bacterial form colonies that differ in size, shape, color, margin, elevation, and texture. These features help in preliminary identification. Second is growth requirement. Different bacteria have specific growth requirements such as oxygen, temperature, and nutritional needs. Observing growth under aerobic or anaerobic condition and on selective or enriched media assists in identification. Biochemical tests. Biochemical testing is one of the most reliable methods for bacterial identification. These tests detect metabolic and enzymatic activities unique to different bacteria. Bacteria are tested for their ability to ferment carbohydrates, produce enzymes, utilize specific substrates, or generate metabolic byproducts. Common biochemical reactions include sugar fermentation, catalase test, oxidase test, urease test, indole production, and nitrate reduction. The pattern of biochemical reactions help differentiate closely related bacterial species. Serological method. Serological identification is based on antigen-antibody reaction. Bacterial antigens react with specific antibodies to form visible reactions such as agglutination or participation. These methods are particularly useful for identifying bacteria that are difficult to culture or differentiate by biochemical tests. Stereotyping is a commonly used in epidemiological investigation to trace sources of infection and outbreaks. Phase typing. Phase typing involves identifying bacteria based on their susceptibility to specific bacteriophages. Different bacterial strains show characteristic pattern of lysis when exposed to phages. This method is mainly used in epidemiological studies rather than routine laboratory diagnosis. Molecular methods of identification. Molecular technique provides rapid and highly accurate bacterial identification. It includes polymerase chain reaction, DNA sequencing, and genomic fingerprinting. Polymerase chain reaction or PCR amplifies specific DNA sequences unique to a bacterial species. It is useful for detecting bacteria directly from clinical samples even when they are present in small numbers. DNA sequencing. Sequencing of conserved genes such as 116S RNA or ribosomal RNA is widely used for bacterial identification and classification. This method allows precise identification and discovery of new bacterial species. Genomic fingerprinting. Techniques such as restriction fragment analysis and multiculus sequences typing are used to compare bacterial strains and study genetic relationships. Automated identification system. Modern laboratories use automated systems that combine biochemical tests, optical detection, and computer algorithm to identify bacteria quickly and accurately. These systems reduce human error and provide standardized treatment. Antibiotic susceptibility testing. Although not a direct identification method, antibiotic susceptibility pattern can support bacterial identification. Certain bacteria show characteristic resistance or sensitivity profile that help in confirming identity. Limitation of bacterial identification. No single method is sufficient for identifying all bacteria. Some bacteria are slow growing, non culturable, or show atypical biochemical reactions. Therefore, a combined method is often required to accurate identification. Conclusion of this lecture video. Identification of bacteria is a multi-step process that relies on morphological, cultural, biochemical, serological, and molecular characteristics. Accurate identification is essential for clinical diagnosis, treatment, and public health management. Advances in molecular biology and automated systems have greatly improved the spread and accuracy of bacterial identification. And this was all about this. Hopefully you like the video. If you do, don't forget to like, share, subscribe, and comment. Thank you for watching.